Welcome back to my YouTube channel, Indian Anatomist. In today's histology session, we'll explore the microscopic structure of a large or elastic artery, such as the aorta, the great vessel that receives blood directly from the heart. We will examine its three tunics, understand how it maintains continuous blood flow. So, let's dive into the beautiful world of vascular histology. Arteries are classified, according to size and wall composition, into three types: large or elastic arteries, medium or muscular arteries, and small arteries or arterioles. The large arteries, like the aorta, brachiocephalic trunk, and common carotid arteries, are called elastic arteries because their walls contain abundant elastic fibers. This elasticity helps them accommodate the pressure surge during systole and recoil during diastole, ensuring a smooth, continuous blood flow. Layers of large artery wall. The wall of a large artery has three tunics: the tunica intima, tunica media, and tunica adventitia. Let's look at each in detail. Observe the relative size of these layers. Let us discuss how the tunica intima is seen in a large artery. Here is the schematic picture showing the three tunics. The tunica intima is the innermost layer, composed of three layers. Inner layer is composed of a single layer of flattened endothelial cells resting on a basement membrane, supported by subendothelial connective tissue. Subendothelial connective tissue is made of loose connective tissue that contains few smooth muscles-like cells called pericytes. At the outer border lies the internal elastic lamina, a wavy, fenestrated sheet of elastic fibers. In elastic arteries, this layer is not distinct, because the tunica media contains multiple elastic lamellae. Now, we will discuss about the tunica media of large artery. The tunica media is the thickest layer, and the hallmark of elastic arteries. It consists of 40 to 70 concentric sheets of elastic lamellae, arranged like a layered spring. Between them, it contains few smooth muscle cells and collagen fibers. There are no fibroblasts in the tunica media, and all the connective tissue elements are produced by the smooth muscle cells. The elasticity of this layer allows the large artery to stretch during systole and recoil during diastole. Finally, how the tunica adventitia looks in large artery. The tunica adventitia is the outermost loose connective tissue layer, composed mainly of collagen and elastic fibers, fibroblasts, and macrophages. It contains vasa viscorum, small vessels supplying the arterial wall, and nervi viscorum, sympathetic nerve fibers that regulate vascular tone. This layer merges with the surrounding connective tissue. Dear students, we have thus far examined the histology of large arteries in a schematic manner. It is expected that all of you comprehend the three tunics and their respective contents. We will now examine the microscopic structure of a real, large or elastic artery. This image depicts the aorta at low magnification 5x. This large artery will now be presented at an increased magnification. At 10x magnification, the inner tunica intima, middle tunica media, and outer tunica adventitia can be readily identified. At this magnification, the endothelium, which is the luminal epithelium of the tunica intima, is visible, as it lines the lumen of the blood vessel. The tunica media represents the thickest middle layer of arteries, characterized by a dark pinkish stain, resulting from circularly oriented elastic fibers, collagen fibers, and a lesser quantity of smooth muscle cells. In H&E staining, elastic fibers are not distinctly stained, however, they can be recognized as numerous faint, concentric, and wavy structures of fibers. The tunica adventitia is the outermost layer of a blood vessel. Tunica adventitia is a layer of loose connective tissue, comprising collagen and elastic fibers, along with fibroblasts and macrophages. Medicos, please refer to the image and its labeling. At 40x magnification, the three layers of the tunica intima can be clearly observed: the endothelial lining, the subendothelial connective tissue, and the internal elastic lamina. The internal elastic lamina represents the first elastic membrane, and is characterized by its delicacy. The tunica media contains numerous distinct circularly oriented elastic fibers, with oval-shaped blue nuclei of smooth muscle cells situated between them. This histomicrograph illustrates part of the tunica media, and the complete tunica adventitia. The external elastic membrane is identified as the external elastic lamina, which separates the tunica media from the tunica adventitia. The tunica adventitia consists of a loose connective tissue layer that accommodates smaller nerves, known as the vasa nervorum, and distinctly visible smaller blood vessels referred to as the vasa viscorum. The vasa viscorum are small blood vessels characterized by a lumen, which is lined with endothelial cells, and encircled by pericytes or smooth muscle cells. The vasa viscorum supplies the entire tunica adventitia and the outer two-thirds of the tunica media. The nutrients from the luminal blood are supplied to the remaining blood vessels via endothelial diffusion. Dear students, I hope you understand the histology of large or elastic arteries. In the next video, I will discuss the medium or muscular artery. If you like this video, please like, comment, share and subscribe. Good day all.